In this video, we will discuss about this T cell mRNA efficiency and activity, or TEA LNPs design using design of experiments. So in this case, what I did is, based on the literature, they divide the components of the lipid nanoparticles in two different phases. First one is this ethanol phases or the organic phase, and second one is the aqueous phase. So aqueous phase contain mRNA, and sometimes they also use ATP, as it has been reported in the literature. If ATP is used along with mRNA, it will enhance the transfection and translation efficiencies of the mRNA. And in the organic or ethanol phase, they have used cholesterol, ionizable lipid, phospholipid, and PEG-lipid. And the mixing of this organic phase and aqueous phase is done either manually through the manual agitation, or it can be done using this microfluidic mixing chip. In both the cases, what they are able to find out, this microfluidic mixing chip results in better transfections and translation of the mRNA. And in order to validate their lipid nanoparticles, the mRNA that they use is of this Firefly Luciferase, so that they can see the signals when the mRNA is been translated. The chemical structures of the lipid nanoparticles are also shown in the panel B, where they have shown what kind of ionizable lipids, phospholipid, cholesterol are used, and the structures for the cholesterol and the PEG-lipid is also shown here. And using this design of experiment, they are able to find out the maxima minima conditions of these two different panels that is shown here. And they started with this A0 condition, and the final conditions they find out is this design space where they use the optimum compositions of the different components of LNPs. And in that case, they can either use ATP or they don't use ATP. And similarly, they did these studies with different initial points that they consider as the panel B, and they come up to the second optimized conditions. So these two optimized conditions is used to find out the final optimized condition, that is design space C, which they considered as a optimized TEA LNP, and that they have used for the further validations. So in that case, in the final conditions, what they do is that they do not consider the ATP. Without ATPs, also they are able to get the better transfection and translations of the mRNA. And the way they divided this mRNA and the ethanol phase is because of the compatibilities, that these cholesterol, ionizable lipids, phospholipids, they together form a lipid nanoparticles, and this mRNA will be inside these lipid nanoparticles. And some results are not presented in this main figure, but they also checked the average nanoparticles or lipid nanoparticles, that are average size of the lipid nanoparticles that are formed, and it comes to be around 100 nanometers for the different conditions. So they finally come to the understanding that the lipid nanoparticles, using their compositions that they have used, will come close to the 100 nanometer, and which is sufficient for the transfections into the cells. And at the same time, the activities, the TEA, in this case, the efficiencies of the mRNA translations and transcriptions, mainly the translations of mRNAs are being reflected here, as they are measuring the translations of the mRNA into the final Firefly Luciferase reporter. However, the activities will be validated once they use the actual mRNA and then use those CAR express or that mRNA expressed T cells against the cancer cells. That's all for this video. I hope you understood to some extent how they use the design of experiment to find out the TEA LNP or T cells mRNA efficiency and activity LNPs. So in this case only the efficiencies are being targeted, as the reporter shows the efficiencies of the mRNA translation. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.